Great, thanks. It's a pleasure to be here. Thanks for the invite to come and present today. So, yeah, this title is perhaps due to the late preparation of this presentation and my just-in-time management approach to life at the moment. So, yeah, we may go off script here. So I think just to go off script immediately, so there are many things that Mihai talked about that I really disagree with quite strongly. And so, yeah, this gives me a perfect platform to say that. So I guess let me just pick some of those first. I thought that maybe you mischaracterized the level of errors, saying there were high errors. I think you need numbers here. I think high and low are kind of meaningless, and one person's high error rate is another person's acceptable error rate. So, you know, when we get to the sequence databases, INSDC databases, if you look per base, the error rates are extremely low, right? So we need to be very specific when we're casting aspersions on all of these data resources, I think. I'll talk a bit more about model collapse. I think that's probably a load of old phooey as well. And this sort of assumes that we have no ways of stopping the training, and the sort of iteratively the whole world gets worse. And I remember papers from the mid-1990s with various people claiming that because we did transitive sequence annotation, that everything would just go to hell and the databases would just be full of nonsense annotation of proteins. I don't think that's happened. I think things have got significantly better over the years. And one example of that, I think, is UniProt, which I know and love. So it hurts me to say that there are spurious proteins in there. They're mistranslations of genes, and they come from, you know, these computational pipelines for predicting genes. But actually, we have a method called anti-FAM that goes and detects them. And over time, actually, the number of, or the proportion of matches to this set of known spurious proteins has been going down. And that's not because we've been removing them. It's because the QC processes have just been continuously improving over time. So, you know, I don't deny that there are many, many errors, particularly in the scientific literature and things which are incorrect. But one of the great things is that we can correct them. We do correct them. Okay, let me just put my soapbox away. Okay, all right. So let's get on with the talk. Okay, so I think it's worth noting that infrastructures are critical, and there are lots of them in our everyday life. The electrical infrastructure, you know, public transport, roads, computer networks, and we all rely on them. Everyone relies on them. No one really thinks about them, right? Well, maybe occasionally they think about them, but it's generally when things go wrong. And then people are shouting at you, and it's pretty similar actually for computational data resources too. And so, yeah, I think it's really good that we are, you know, constantly trying to remind people of just how important these infrastructures are. It's funny, so, you know, over the years, I've been involved in databases like PFAM and UniProt and InterPro and so on. And I get quite a lot of mails from journal editors asking us to submit papers describing our resources to them. So it seems like everyone wants to publish our papers. It's just and recognize how important these things are, but no one wants to pay for them. Okay, so that's the big trick is working out how to, you know, demonstrate our importance. Okay, so yeah, we're talking about computational biology infrastructure. I think here we are really focusing on data resources. As Mihai mentioned, there are also, you know, software infrastructures that are really critical for all the work that we do, so that we don't all go around reinventing the same bits of code, and we try and share that. The computational infrastructure, of course, absolutely critical. I think we don't have to worry about that too much. That with the rise of AI, it's not going to be too long before much of the planet is covered in data centers, and, you know, we have a lot of compute available. But we shouldn't underestimate the importance of data standards as well, because that's what really allows the whole ecosystem to work. Without that, it would be, let's see, even more chaos than it kind of is currently. And then we shouldn't sort of forget about the importance of training materials, universities. All of that stuff is important to support the computational biology environment. Okay, so yeah, I'm from EMBL EBI, which is a world-leading source of public biomolecular data. I'm part of the EMBL laboratory and based not so far from here in the UK. Actually, distance is relative, so for me it's a really long way, but for the Americans in the audience, it's like just next door. Yeah, just a couple of 100 miles away. Other products are available. And so, yeah, there are these other places that do similar kind of things. NCBI, of course, the China National Center for Bioinformation, Swiss Institute of Bioinformatics. I'm sure there's many other places as well that provide important parts of that infrastructure. And the whole is greater than the sum of the parts, I think. So if we zoom in on EBI, so here we have about 40 different data resources across the range of different types of scale, from small molecules through genes, proteins, up to sort of tissues, disease, and through to the literature as well. My sort of interests are in the protein area, so PFAM, UniProt, Interpro, RFAM and RNA Central are the kind of things that I'm involved in the management of. So any questions on that, really happy to take them. So I did a screen grab from this great database called Database Commons, where based in the China Center of National Center for Bioinformation, and they record almost 7,000 databases now. It's really remarkable just how many there are. And I think most of us thought there were about 2 or maybe 2,500, 3,000, but things have moved on. But, not all databases are built equal. I think it's important to remember that there are really critical bits of infrastructure, and you're going to hear from GBC and I think Elixir too, who have ways of recording which are the really important ones and get a nice badge to say so. Yeah, I mentioned that these databases are highly cited, so Database Commons provides this nice, highly cited ones. I was really gratified to see that a couple of my favorite databases make that list, so that was nice to see. Okay, before I go on to the next slide, I'm just going to grab a drink. Just a second. Much greener. Okay. Okay. Yeah. So now I want to talk about curation, and so this is really, I think, at the heart of the knowledge bases. So, as Mihai said, we have these sort of data resources where you submit data from the submitters, databases that sort of integrate things. And we have the knowledge bases where there are real people who care about the data and are trying to improve it, maybe selecting what data goes in, what doesn't go in. And curation absolutely remains critical in the age of AI. I think there's the risk of a perception that, as ChatGPT, Claude, all these things get better and better, that you will not need curators. This is absolutely not the case. These models, the data, they have huge teams of people who've checked data, getting good data sets and pulling them in. And it's been really critical for the development of AI itself. And particularly for supervised machine learning, you need a ground truth. This is absolutely critical. And this comes from high quality curated data sets. And I think if it doesn't, then this is where we sort of start heading towards model collapse. And I think many of those ideas do come from the idea of, oh, let's just let the AI sort of retrain itself, and that can go bad. I mean, there are great examples where, if done carefully, this sort of bootstrapping approach to ML does work very well. And AlphaFold is a great example of that. So in John Jumper's talk, 75% of the data they trained on was actually generated by AlphaFold itself. I hear very often in these sorts of presentations that curation is not scalable, that we're drowning under a deluge of data. There's a lot of data, but in terms of the scientific literature, it's not growing super fast. The complexity of the papers, I think, is getting bigger, and people are putting more information in the supplementals. So it is challenging, but I think curation can scale because we develop new tools and infrastructure, and now curation is becoming, I think, turbocharged by AI. Through careful application of these models, we can ensure high data quality. That's not to say that we wouldn't love a little bit more money for curation, right? We could definitely do more. But even with the resources that we have with AI coming along, I think there are real opportunities for expanding our ambitions about what we can do. And certainly with PFAM, this database of protein families, I've been working on this for 25 years now. I thought we'd be done by now, but apparently not. But we have 25,000 families that we've built over those 25 years. But with AI, we're speeding up, getting better and quicker. And honestly, I think we'll essentially be done with protein family domain curation in the next 5 years because of that. Okay. Well, no, but then I want to go on to the next problem. Yeah. We need to work out what the functions of all these proteins are, how they interact. There are so many more good things to do. And honestly, there'll be some refinements, and there's a long tail of small protein families that some people may care about. But essentially, we'll have the large bulk of that task done. Okay, so yeah, curation, I think, does help stop this model collapse. One thing, another sort of thought about AI is that at the moment, at least, it's rather uncritical. You can pump papers into ChatGPT and it will extract the knowledge in some form or other, but it can't make judgments about how good the experiments are. And one of the great things about the UniProt curators is they read the papers. They're all PhD level scientists, and they go, yeah, I don't think that really supports these conclusions. And so that data will not go in. And I think that's really important. And, I can imagine in the future that maybe AI can extract everything from the whole literature. We can have a UniProt entry with everything said about P53. It would be a nightmare, honestly. It would have so much trash in it. It would be unreadable and hugely long. So be careful what you wish for. So Mihai, I think, already said this, that knowledge bases, that they're changing over time, and the state of our knowledge really is changing, and what we thought a few years ago may not be the case. And so that's one of the reasons why knowledge bases are so important, because in the sort of the archival resources, you do tend to freeze some of that incorrect knowledge and it just gets stuck there. And finally, on curation, so community feedback is really important. We get to all of these resources, we get kind citizens who tell us when things are wrong. They don't always do it, which is a bit of a shame, but we really appreciate it when they do. There have been thoughts, and Amos mentioned this in his keynote, that in the 2000s was the era of community curation was going to solve everything. Community curation is an extremely challenging thing, and very few examples of where that's been successful. And when you really dig into the more successful examples, it tends to be the team that produced the resource that are really doing that community curation, and maybe one or two really engaged people. So I wanted to mention about resource life cycles. So of these 6,975 resources, actually, many of them are not taking any sort of effort or funding. They may be defunct, stopped. They may be, there's a website still hanging on using some old operating system on a server that might go down anytime. So I think quite a large number of those resources, are sort of very small projects, maybe one-off lab things out of a research project. So I don't think that the number of really important resources is anything like 7,000. So I sort of think of the life cycle of resources in this way. So we have, the excitement of the resource being born, everyone's very enthusiastic, puts things together, gets the data in. But there's quite a hurdle here. And so I think in the research sort of field, being able to produce this data once is enough. You publish your paper. But then there's a sort of a whole, people like myself and many of you in the room who are service oriented, and you don't have to just get this data once, you have to release it on a regular basis. So for me, that's sort of maturing. If you're able to, persuade funders to fund this resource and sustain it, then I think you're sort of moving into a more mature state. Over time, many resources move into what I call a sort of senescent state, where maybe the funding's gone away, the student who put this together goes away, and then it's maybe sort of hanging on. And actually, very few databases seem to ever retire or go away. They just hang around like I'm not going to say what they hang around like. But yeah, they just never go away. It's really difficult to kill a database. They're like the undead. Yeah. So the resources that do retire, they tend to be at the larger centers where they have an organized life cycle. And so, that's another reason why these places are so important. Okay, so I have little time left, so let me whiz through these things. So leadership and succession. So successful resources generally have a really passionate leader, the person who thinks this is important, right? And pushes to get the resources up and running. Those leaders sometimes retire or move on to other projects. And this puts the project at risk. And if it's critical infrastructure, it's going to be a bit of a disaster. So it's essential that you have succession planning. And UniProt is a great example where we had Amos and Rolf Apfeiler forming SwissProt Tremble. They're no longer involved in the project. And actually, there's been a series of PIs with Kathy Wu coming on from PIR. And so over time, that's been really critical, and it's brought in new ideas over that time. And then finally, so what happens when these people retire? Well, sometimes the resources kind of get rescued. And so I like this example. So InterPro is a collection of protein family resources. There are 13 of them. Actually, 2 of those now, Prince and SFLD, the PIs for that retired, and the database is closed down. Fortunately, we were able to persuade those people to give us permission to relicense the final releases in CC0 format. And now we make those available through InterPro, and we run the models so they're still there and searchable. So these sort of umbrella resources can act as a kind of a data archive, maybe not in perpetuity, but in the much longer term. And even here we have some of these more senescent resources where they're really not developing right now. Okay, so I think we come to an abrupt end there because I ran out of time preparing the slides. So thank you very much for your attention, and I'm happy to take questions at the end. If there's... Okay, great. Thank you very much for your attention. Thanks.
Great talk, Alex. Thanks for summarizing this up so well and so cleanly. I have a question about curation.
Mm-hmm.
So I think the average time between a paper getting published about a certain protein function and that function entering UniProt is 4 years, but the median it can be much longer. It's a very long curve distribution. It is 4 years. We checked.
Wow, it would be great to see the data.
We'll show it to you. Yeah, it's 4 years. My thought is, what do you think about having some sort of mandate for the authors to at least suggest, Go terms or whatever ontology is being used as part of the curation to speed up the curation process?
So this is sort of the community curation part. So we do have a community submission to UniProt. So on every UniProt entry, you will find a link for community submission where you can you can submit information of paper relevant things. To do that, you authenticate against your ORCID, so we know we have good provenance of who is doing that, and we check all of those entries.
Yeah, that's voluntary, but I'm thinking of like mandating, like if you have to put in a sequence, you also have to put in a function, if that's what you're dealing with.
Good luck. So, I mean, just related to that, there are lots of people who've suggested these sorts of things over the years, and it never works. And honestly, so people think that anyone can be a curator. Oh, yeah, there's some person from a lab, they're a wet lab experimentalist, that they can curate the data accurately. This is not true. I would dare them to say, oh, let me go into their wet lab and do the experiments. Curation is a proper job, and it takes training and time to do it properly. Thank you.
Yeah, thank you so much, Alex.